Good morning. It is Monday, December 14th. We're going into our last full week before our holiday break. And today we're going to start working on something that for most of you should be a bit of review. We started talking about genetics last week and working through it. Today we're going to start our work with Punnett Squares. So with that said, welcome back. All right, so here we are. We are in Unit 4 for genetics that we started last week. And now we are in week number 2. Now, here you can see what we're going to be working on for Monday and Tuesday of this week, our notes in the genetics PowerPoint. I've included the second one, and it's just to help people who may not remember exactly how to do Punnett Squares. This will help you walk through step by step how you do it. So please, if you have any issues getting the Punnett Squares done for our worksheet today, then make sure that you check this extra help Punnett PowerPoint. It will help you, I promise. All right, so moving on into genetics. I don't know if you remember, but we ended last week talking about Gregor Mendel. Remember, Mendel is one of the 2 scientists that you're going to need to know the name of. The other one will, of course, be Charles Darwin. Mendel is our father of genetics because he actually figured out the patterns of inheritance way before anybody else did. Like 80 years before anybody else did. The fact that everybody ignored his paperwork does not mean that he didn't get it figured out. It just means that nobody was listening, which is really too bad. So let's review some of those early terms that we went over that I had everybody write down. So at this point, what we're going to move on with for this is we are going to continue with the path of research and discovery that happened real world at this point, which means people did not know what DNA was. We couldn't figure out what the molecule that passed on traits were. So instead, we looked at the pattern. Can we figure out the pattern? And if we can figure out the pattern, then at least we can have some answer, some explanation for how things work. So remember our gene, our trait, and our alleles. You should have all of these written down. So the example, the gene is for eye color. The trait is going to be whatever the normal color of the irises of your eyes are, but each individual might have green or blue or brown or gray or different alleles, different versions of that gene. Now, what's really interesting is that if we compare living things, we see that so much of our DNA is shared with everything. Huge amounts that you might never even anticipate. Because of that, genetics, DNA, all of that is often referred to as the unity of life because we are all connected. We have all evolved from the same original common ancestor going back like 3.8 billion years. And our proof is in our DNA. Even with something as different as yeast, we share 26% of our genes. With something mammalian like a mouse, we share 92% of our genes. And obviously, our closest living relative, chimpanzees, we share upwards of 98%. So we share a lot of our genes with everything else on this planet. We are all, to some degree, the same thing. All right, so let's talk about how we figure this out. So when we take a look at a gene, what we're going to be looking at are the 2 alleles that you get for every gene. All right, so the way that we term it is you get 1 allele from mom's egg cell, you get 1 allele from dad's sperm cell. When the 2 of them come together, that means that your genetic code is 1 + 1, so 2. You get 1 letter from mom, 1 letter from dad, you have 2 letters. So our genetic code will always have two different allele letters, always. So here is our example. We get a capital B for brown hair from mom. So we get a dominant allele, which we term capital B to show that it's dominant. And we get a little b for blonde hair from dad. This is a recessive allele. So we use a lowercase letter in order to show that. So our gene, our total alleles are big B, little b. Does it matter what order these are in? No, it does not. Because when we look at this, due to dominance, your hair will be brown because the dominant capital B will overpower the recessive blonde hair allele. We carry that recessive blonde allele, but we're not showing it. It's been overwhelmed by the big, powerful, dominant allele. You guys remember that idea? Now, this introduces two super important terms that we're going to see all over the place. Number one, heterozygous. Number two, homozygous. Now, once again, you don't need to know what all of this means. If you remember that prefix from the Latin, it gives you all the information that you need. Hetero means different, which means we've got two different alleles. We have one dominant and one recessive. Homo, Latin for same, which means both of our alleles must be the same. So little b is homozygous. Two little b's are the same allele. Or capital B, that would be homozygous dominant. Our first one, little b, would be homozygous recessive. So homozygous, the same two. Heterozygous, two different ones. It gives us a really powerful way to explain and talk about these alleles and the genes that individuals have by saying dad is heterozygous, mom is homozygous recessive. And now you should be able to figure out what the letters would be. Here's a hint. It doesn't matter what the letters are. You literally pick any letter that you want for any one of these. It doesn't matter if it matches with the trait. You will see that in most cases, as we move forward, I will use B's. And the reason is because the capital B and the lowercase b look very different. So it's easy to tell what the dominant form is and what the recessive form is. If I used S, that would be a lot harder to tell because capital S or lowercase s are going to look basically the same. The only difference is a little bit of size. So I always suggest, when in doubt, choose letters that the capital and the lowercase look very different from each other. All right, so using this information, if we have a gene, right, and this is the gene for something like eye color, realize that your gene is actually built of two different alleles, in which case this would be heterozygous. Now we could line up your entire genetic code like this, where what we do is we just take the two alleles that you have for each one and set them up. So we see that you are heterozygous for this gene, heterozygous for letter D, homozygous recessive for letter E, and heterozygous again for Q. That would just be your genetic code. Seems like the lights are going to turn off on me. And that will bring us to our Punnett squares. So these are super, super easy. And if they challenge you a lot, make sure you check. There goes the lights, of course. Make sure that you check the slideshow that I put on for extra help and get some practice with them. And if you need more, let me know. I can provide that for you. I'm going to get the lights back on because otherwise I'm like, I don't know, a ghost in the darkness. Yay, technology. All right, so it is good to point out Punnett squares are designed only to predict genetics. Make sure this does not tell you what you're going to get. This tells you the most likely mathematical result. So we make this box. We put one line down the middle, one line across the middle. So we get 4 roughly equal boxes. And then we're going to get our alleles. And you're going to be told to some degree what the alleles are. So dad is capital B, homozygous dominant. Mom is capital B, little b, heterozygous. So we're going to take those 2 alleles and we're going to spread them out. So dad's 2 alleles, one drops down here. The other capital B splits to go over here. Mom's side, capital B comes to one, little b moves to the other box. Why? Because when we make sperm or egg cells through meiosis, remember they each end up with half of our genetic code, which means that they are going to get one half or the other half. So we separate them. Then you're going to take dad's alleles and you're going to push them down. Copy them in each box below. You're going to take mom's alleles, push them over. Copy them in each box. So this one is capital B, homozygous dominant, because we got a capital B from dad right here and a capital B from mom right there. And those 2 got pushed together into that box. So you'll notice there's a capital B here and a capital B there. That was pushed down from dad. Capital B here, capital B there, pushed down from dad. Then mom's push across. So we add a capital B here and a capital B there because that's what she had here. And finally, in this bottom row, this little b gets pushed across. So we get a little b here and a little b there. So these are all the possible combinations that these 2 individuals can have. Now, if we looked at this gene and we said this is eye color, and capital B is the dominant for brown, little b is the recessive for blue, what color eyes would these children have? Brown. All of them. Because they all have at least one dominant allele. All right, here's another one, right? We're saying here the gene is once again eye color. Capital B equals brown, little b equals blue. Dad is homozygous recessive, little b. Mom is homozygous dominant, big B. And so you push dad's alleles down, push mom's alleles across. Every one of them is heterozygous, capital B, little b. What color eyes will they have? Brown or blue? Blue. Brown, because they all have a dominant. Now, that will bring us to probability. They're never going to ask you what amount of them. They're going to ask you what is the probability, what percent of the offspring. So here's an easy thing. We have 4 different boxes, right? If I take a pizza and I cut that pizza into 4 equal slices, how much is each one worth? Each one will be worth 25%, 1/4. All right, I hope that helps. I've got somebody at the door, so I'm going to take care of that. Ladies and gentlemen, I look forward to seeing you in class.